Yes,
We can
yes.
start? Yeah, let's start. Okay, today we are welcoming our guest speaker of the African Pharmacogenomic Network webinar series that's scheduled on Thursday, every month on Thursday. And today we are inviting our very important speaker, Professor Ozlem Bishop. She's a full professor in structural bioinformatics at Rhodes University in South Africa and distinguished adjunct professor at Zavida University in India, Chennai, India. She received her Bachelor of Science degree in physics from Bangazi University, Istanbul, in Turkey. And she moved to the Department of Molecular Biology and Genetics at the same university for her Master of Science degree. She obtained her philosophy doctor from Max Planck Institute from Molecular Genetics and Free University in Berlin, in Germany, in 2003. While doing her PhD, Ozlem became interested in structural biology, and during her postdoctoral positions in Texas University in USA, University of Western Cape Town, and University of Pretoria, South Africa, she gained experience in structural bioinformatics as well as structural biology. In 2009, Ozlem took up an academic position at Rhodes University in South Africa. She established her research unit in bioinformatics, Ruby unit. In 2013, she has graduated 27 PhD and 40 master science students since she joined Rhodes University. Rhodes University Vice-Chancellor Distinguished Senior Researcher Award. She got that in for 2020. And South African Society for Bioinformatics Silver Award in 2022. Ozlem's broad research interest is structural bioinformatics and its application to drug design and development. Her recent interest is in the allosteric mechanism of proteins and understanding the effects of non-synonymous single nucleotide, variation on protein structure, and function in the context of drug resistance and drug metabolism. She has published over 100 research articles, and she's like a brilliant researcher. We can talk more than that. Okay, today we're welcoming Professor Ozlem Bishop, and now the floor is yours.
Thank you, Yasser, for a very kind introduction. Hi everybody, I'm sorry I will not start my video because I did so 10 minutes ago, everything crashed. You might see me at the end when I dare to just switch on my video. Okay, it's a great pleasure for me to be part of this webinar series. I would like to thank organizers for inviting me. Today I will be talking about some of our recent research and publications actually, which are all related to the protein analysis. But before starting, let me introduce us. We are research unit in bioinformatics at Rhodes University. I'm not sure if you are seeing my mouse cursor. We are located in South Africa in a small university town called Makanda Grahamstown. Our research interest is in structural bioinformatics and its applications to disease and health-related projects. Structural bioinformatics in general focuses on the identification and analysis of structure, dynamics, and interaction of biological micromolecules in 3D space. In that respect, we are mainly focusing on protein structures. It brings the information from sequence and structure together to deduce functional aspects in micromolecules. And the structure is particularly important for computational and structural drug discovery, of course. So if you look at our research, it can be perhaps grouped under four themes. These are the drug design and resistance, rare diseases, pandemic preparedness, and of course, drug metabolism and pharmacogenomics. So our research is on analyzing the protein structures in a dynamic manner. We do lots of molecular dynamic simulations, as you can see on the left corner. And the reason why we do that, we would like to analyze structures under a cell-like environment in native states. So our overarching aim in all our research is basically to understand the mechanism of the causality of the problem at the atomic level. And the problem can be drug resistance, or the problems in drug metabolism, or disease itself, or the pathogen evolution. So regardless of this, we want to understand the causality of it at atomic level. And we want to do this to basically design drugs that are long lasting, more effective, and safer. So we all know that drug research and development is a very long process with lots of challenges. And one of the challenges is the high attrition rate. As you can see here, when you move from lead optimization to preclinical test and further, the decreased attrition rate is very high. So it means that certain things are not done correctly in basically in the first couple of steps. Even we get the drugs at the end and goes to market, we still face number of issues on marketed drugs, such as drug resistance, adverse drug reactions, and lack of efficacy. If we look at these problems, they are basically due to the mutations or variations. Mutations are drug resistance because pathogens basically mutate under the drug pressure and gain drug resistance. And adverse drug reactions and lack of efficacy is due to the variations on different human populations. So this, of course, is important in the African context because in Africa we have lots of diseases, and these diseases disproportionately affect African populations. And there is very limited drug development to these diseases. And then on the other side, whatever we develop against these diseases, we face to drug resistance. And the circle between drug resistance and replacing these drugs with the new one is not there, because drug development is much slower than the pathogen gains resistance. So one way to handle this challenge is perhaps to design drugs capable of bypassing pathogen resistance. But of course, this is not the only thing that we need to consider. We also need to consider, especially for the African populations, the genetic diversity, because African continent hosts the most genetic diversity in the world, and in the world, and the diversity is not well studied. It is not well studied in the clinical side, but also it is not considered while designing these drugs, even for the diseases very specific to Africa. So at the end, we end up with medications that are not optimized for African populations, which can cause a sort of toxicity in people. So 20 to 95% of variability in drug response is basically actually due to human genome genetic factors. So in that respect, it is highly crucial to understand how the variations in our genomes relate to the certain drugs. So we know that when we administer the medication to people, people can show different effects, and there are four scenarios here. For instance, like desired response, but with lots of adverse drug reactions or not effective for the disease, and also you get adverse drug reactions, or you don't see any effect at all. But what we want is basically here, we would like to have desired response to disease with no adverse drug reactions. And so it means that for the people for the same diseases should definitely not receive the same treatment. So these people need to be actually categorized according to genetic variations, and optimal the doses should be applied to the individual populations. But this is surely very costly to do it, especially for the African populations. So what we propose to do in our research is first to understand underlying molecular mechanisms of the effect of missense mutations on drug metabolizing proteins at atomic level, and then use this information to predict the effect of the variations with unknown clinical implications. So if we can do this, then we hope that we can accelerate this process with less costly ways. So now, understanding the variations or the mutations in protein level, it is not always a very easy job. If the mutation or the variation is the active site, orthosteric site, functional site of the protein, then it's fine. We can really easily understand why the protein is not functioning as we hoped it would. But most of the time, the variations in pharmacogens are away from the active sites. And these sites are called allosteric sites, where these variations located. And yet they, even though they are far from the active site, they still somehow affect the function. So and the other complication is, of course, sometimes we get not only one or two mutations in proteins, number of them, and they collectively work and change the function of the protein. We actually, a couple of years ago, published a review article explaining all these mechanisms. So I like to give this analogy for the allosteric communication. It's like a Chinese whisper. You can imagine that these are each individuals are like residues in the protein. You give input from one side of the protein and you get output from the active side. And this is how a normal protein function. There is continuous communication between the residues. But sometimes something happens on the way into one of the residues, like here. What happens if there is a person with hearing impairment? The whole communication changes. And that is basically what is happening with the variations in the proteins. The communication changes. So in that respect, we thought that we can actually analyze the proteins in a way like networks, communication networks, and we can look at the changes in these communication networks if there is a mutation or variation in certain residues. So we carried this kind of networks over the MD simulations, and we call this dynamic residue networks. So the tools are published. There is a standalone tool, and more recently we established this approach in a web server base. And what we do, we now have a, let's say, a protein. We calculate the network. When you know the network, how the residues interact with each other, then you can actually retrieve many different information by using different metric calculations. My three favorite network analysis metrics are the between centrality, BC, closeness centrality, and eigenvalue centrality. So when you calculate BC, basically you can identify the residues highly used in the communications. So this is basically the residues passed information, the most frequently passed information from one side to other. Closeness centrality basically can be defined as point of rapid diffusion of information, looks at the individual residues with more communication than any other residues. And eigencentrality looks at the residues connected to many residues which connected to many more. It's like, and these actually definitions are coming from social sciences, and we can say that EC can be defined like important famous residues connected to more important famous residues. So that basically defines the residues which connected to more functionally other residues. So knowing that now with all this information, I would like to give you two examples. The first publication, both of them came this year. First publication that I would like to talk is NET2 enzyme. Um, and NET2 is important for I think to understand for in the African context, because this enzyme acetylates the isoniazide, and it has two different forms of the enzyme depending on the allele. It can be a rapid form or the slow form, slow acetylation. The terminology is a bit unusual in NET2 community. Normally, pharmacogens are divided into normal functioning, slow functioning, rapid functioning, but in NET2, there is only two division, rapid or the slow. This work is done by my postdoctoral fellow, Dr. Wade. And what we did here is basically looked at number of alleles, 7 alleles. But before coming that, let me just introduce the protein. Protein is very small. It has 290 residues, though it has three domains in this small form. Domain 1, domain 2, domain 3, and which has also interdomain region in between, and a mammalian insert specific to mammalians, human. And the active site is in the middle, and it's actually covered with the C-terminal end. And it has also active site loop, which is part of domain 2. What you are seeing on the right side is the catalytic triad in cyan and the potential putative isoniazide binding residues, which are shown in pink. So the reaction mechanism is presented in this figure. I'm not gonna enter into details, but basically what it happens, Acetyl coenzyme binds to the enzyme and transfer the acetyl group to the substrate. On the right, you see that the isoniazide is a substrate and acetylate the substrate, and that is the key function of this enzyme. And this reaction, depending on the allele, can be fast or slow. And if it's slow, of course, it causes problems. When we started this project, we used this nomenclature, but on the way it changed. But nevertheless, we had two rapid systems and five slow systems. And slow systems, there are also some, in some cases, synonymous changes and some changes in introns, but of course we only looked at according to the non-synonymous changes. So when we published the articles, this this was the end nomenclature. They are still changing in the transition of changing the definitions. We, as a reference, we wanted to use the protein with K in position 268, and that was the reference. Now they change it to another reference where this position has R. But it didn't matter for us. We still used legacy reference K268, which was the rapid, had rapid phenotype, and the current reference has also rapid phenotype. Okay, what I would like to show you here is, please look at the variations. They are in spheres. They are, as you can see, all of them are away from the active site. There is only one D122, which is in system 5, net to star 48 allele has the mutation in the active site. The rest is away from the active site. The methodology that we used involved molecular dynamics, three independent MD simulations for reference, and also for the new reference that we did, and the one MD simulation for each of the variants. So our reference, as I said, K268 system. And what we did, we calculated, we did some post-MD analysis, and then compared to two rapid systems. And then we wanted to understand how the shift is from rapid to slow, because slow systems basically included only one or two additional variations to this rapid system. So we wanted to see how actually additional variations shift the system so drastically. And these simulations, three replicates are here. The red ones are the allele systems. As you can see, I mean, everything seemed more or less stable. We didn't see any drastic changes in these systems when we look at globally. And you can see root mean square deviations in Weiland plots. I mean, if you don't know what root mean square deviation is, basically we compare the structure to initial structure over the MD simulations and see how the structural changes. And as you can see in the reference systems, three reference systems, and the other rapid system behaved more or less the same. We saw some slight variations in one system, but for the sake of the time, I'm gonna only focus on these three systems. Okay, as a first thing, we analyzed our reference systems. We looked at root mean square RMSF, which is root mean square fluctuation. Basically, what it means is how the average position of residue is changing over the MD simulations, if it's fluctuating or not. And so we identified which regions are fluctuating the most. There are lots of details in the article. I'm not going to enter any of them because it's really a lot, basically. But the red color basically the brighter red color shows the highly fluctuating regions in our reference. The next thing we did is basically we wanted to see how the variation in this position, if A moves from K to R, would change the structure, because both of them, either K or R in that position, they actually functioning as rapid acetylators. And as you can see, what you are seeing here is basically a root mean square deviation subtraction between two systems, and the gray, I mean, the blue shows more rigidity, red shows more fluctuations in this system. And as you can see, there is hardly any changes. So both of them are basically showing the similar behavior. Hydrogen bonding, then we identify the hydrogen bonding in our reference. We saw that the highest hydrogen bonding is in the 1,2,2 residue, which is active site residue, followed by R64, which is also highly important residue. And soon enough, we will look at the allele where there is that there is mutation variation in that residue. We identified some slight changes in the hydrogen bonding between two repeat systems, and the significant ones are actually presented as dots, and these significant residues are identified by three standard deviations. If it's higher than three standard deviation between two systems, then we mark them. Between its centrality, we identified a kind of communication path around the active site by looking at network analysis. And when we compare to the other rapid system, again, this is delta by subtracting the residue values from two systems. We are seeing what is changing. We saw some slight reduction in the active site in certain active site residues, or around active site residues. And we also saw some changes in this should be EC, eigencentrality and closeness centrality. But overall, there was no really, even though we were identifying changes, there was like only one residue significantly different, and or two residues in EC. So overall, we could say that these two rapid systems were behaving, at least for these metric systems, more or less the same way. As a next thing, what we did, we wanted to see how the shift is from rapid systems to slow systems by looking at the same matrices. So RMSF, hydrogen bonding, and etc. And we always use delta to be able to see the deviations. So what we are doing is we have three runs, MD runs from our reference. We average it and then subtract from each other system, and then identify the top and bottom X%, in this case 3% or 5%, and then from there we really wanted to see significant changes by looking at three standard deviation. So what you are seeing as X is the significant changes with respect to the reference. And as you can see here in this system, which basically has one additional variation, R64 Q variation, shows significant fluctuation in domain 1 region and here in inter-domain region as well as in C-terminal. And this is all mapped here. So there is significant change in the fluctuation here. If we look at this system where there are now one more additional variation on top of this system, then we see additional fluctuation is coming here. And please note that the variations are, in this case, this variation is again far from where these changes are happening. And D112 variation is the reason why I'm presenting this. This is the only one where the mutation is in active site and would directly affect the metabolism. So in the same way, when we look at the hydrogen bonding, we again see similar things. I'm just gonna very quickly go through without entering the details, but you can see that there is reduction in the hydrogen bonding in domain 1 area, where basically the mutations are. Actually, it is known that in this mutation, R64Q mutation, there is a hydrogen bond loss between these two residues, which leads to protein thermostability. This reduces the thermostability. This is known in the literature. If we look at now one more additional variation, I114T, we see that there is further reduction in the hydrogen bonding in another region, which is near to active site actually. Interestingly, what we see is in this variation, D112N variation, where the mutation is in the active site, and the whole hydrogen bond system changes. There is really lots of reductions and regains of the hydrogen bond. So it is more impactful because as you might remember, the highest in the reference structure, highest hydrogen bonding was done by this residue. So the mutation on this one changes everything in the structure with respect to hydrogen bonding. So the BC, we see again the communication path changes, but I think the most important change we see again in D112N, and again the strongest change it seems in the one where the active site residue variation is, but we also see changes, dramatic changes, reduction in the CC values in this variation. And when we look at the EC, many residues, again, around the catalytic active site had a large decrease in D112. So what I would like to say here that we see more dramatic changes if the residue mutation is in the active site, more subtle changes if it's allosteric mutation. So now when we put all these things together for each variant, for this variant here, R64Q K268R variant, you can see that there is hydrogen reduction in domain 1 area, which leads to basically increased flexibility in the same region, and quite a bit of changes in the network communication path, like D122 active site catalytic residue loses the between centrality values, and many other things happens, and also distances changes in the active site in some residues. So if we look at this one, this variant, if we put all the metrics analysis together, again, we see that 64Q causes this reduction of hydrogen bond, which causes the RMSF values, the flexibility to increase, and on top of that, introduction of I1140 mutation cause additional change, hydrogen bond loss in another region near to active site, which leads to additional flexibility. And this causes, of course, quite a bit of changes in closeness centrality. All the network behavior changes here. So, and the third one, which we have basically we see drastic changes in the hydrogen. The flexibility is not the issue. It doesn't affect really the overall flexibility, but it changes the communication paths and the hydrogen bonding. So basically, we see different mechanisms depending on where the mutation is. So overall, what I would like to say here that yes, with our approach, we can see that two rapid systems show very similar behavior in the metrics that we calculated. But then when we look at the slow acetylate and the slow systems, we see dramatic changes, but very local allosteric effects. And this is basically called actually dynamic allostery. It doesn't affect the mutations, doesn't affect the whole conformation, but the local cause local changes, structural changes in the protein. So we actually observe this kind of behavior also in cytochrome P450 systems. And in the rest of my time, I think I have still 5, 6, 7 minutes. I will be talking quickly about our second article, which was published this year in a special issue in JMB. And this work mainly was done by my PhD student, Rehema, with input from Wade, Ellen, and Chichi. Here we looked at CYP3A4 alleles. This protein is like many other cytochrome proteins. It has the same fold, same structure with the 14 alpha helices for beta sheets. But the important thing with this protein is that it metabolizes roughly 50% of FDA approved drugs. So it is in that respect, it is really important to understand how the alleles are affecting the behavior of this protein. And the challenge with this protein, depending on it's not like a black and white, it's not like slow or rapid metabolizer. One allele, it depends on basically the drug. Depending on the drug, sometimes allele behaves like fast metabolizer and sometimes slow metabolizer. So it has a number of unique features, even though overall structure is very similar to any other CYP enzymes. And I'm just very quickly mention three of them. It has hydrophobic region here, which is linked to the membrane. It has shorter F and G helices than any other CYP enzymes, which is important because then it exposes the active sites more. And this is basically perpendicular to I helix, and I helix is extremely important in the function of the enzyme, especially in the binding, substrate binding mechanism. And this protein also has phenylalanine cluster, which is on top of the active site. It's like gatekeepers in the substrate entrance to the channel. And so we looked at, this is very polymorphic protein with ethnic variations. We have 26 allele. In this article, we looked at specifically 13 alleles, which we see in African population. I would like to just emphasize that I'm gonna focus mainly this one, STAR3 allele, which is near normal metabolizer, and STAR32, which we know that it's either fast or slow metabolizer in many different cases. So this is a methodology. This is our graphical abstract, but summarizes all the methodology that we used. We did a delta analysis, as I showed in Wade's article, but we also looked at active site, heme analysis, channel analysis, and some additional network analysis. So the behavior of these proteins were more or less same. Two of them showed different two conformations. This is the STAR3 allele, which was very similar to reference protein, and this is STAR32 allele, which basically consistently show little bit higher RMSD than the reference systems. When we look at flexibility, and we did this in many different ways, we use normal mode analysis, delta calculations. What you are seeing here is some systems, the GH loop area is getting very flexible compared to the reference systems. Okay. On the other hand, STAR3, you can see that it's more or less behaving like normal systems. Okay, so this GH loop, which is around here, and I circled the systems where you see this increased flexibility, is around here, which is basically where the short two helices are, and basically the flexibility, and the mutations are far from this region. Actually, again, we are seeing allosteric behavior. So when we look at the active site analysis, when we look at the active site, the reference protein, the most flexible residue for the reference protein was phenylalanine 215, which is part of this phenylalanine cluster, which is a sort of gatekeeper in the substrate channel. And what we observed is actually in number of cases, this residue became more rigid. So it, in a way, might explain why they might have reduced function in certain cases. We also looked at heme behavior. I'm not going to go into details, but as you can see, the interactions are more or less same. So heme is staying stable, but the things around the heme is changing. One striking result came from hydrogen bond analysis, and I'm gonna focus only here P32 star allele P32 system. As you can see, as you can see with these yellow arrows here, this system gained two additional hydrogen bonding, and this hydrogen bonding was between these two residues, 212 and 304 and 250 and 292, which is shown here. But these two additional hydrogen bonding basically cause the bend in the I helix. As you remember, I told you that the I helix is very important, is in the whole activity of the protein, and the variation is again far from where the band is, because band is near to as you can see, near to the heme area. Okay, so that was quite interesting and could explain us how the mechanism could change drastically in STAR32 allele. So ranking the alleles by total hydrogen bonds, as you can see here, what was very interesting, we have these three references, and STAR3 allele is the closest hydrogen bond behavior to that, and we know that that is near to normal functioning allele. On the other hand, the farthest, the highest change was in STAR32 allele. Okay, overall, in conclusion, we could show the actually differences, significant difference between STAR32 allele, which is known with the reduced function in binding of like quinine and ibrutinib metabolism, or increased metabolism in other drugs. On the other hand, STAR3 is known as near normal metabolizer, which has no effect in these two drug metabolism, and increased metabolism in some others. So basically, I think our findings can more or less range the star alleles according to their level of fast or slow metabolization. And we think that we are on the right track, and with more analysis, we will be able to find a more generalizable approach that we can apply to the alleles with unknown clinical behavior. So with that, I would like to finish my talk and thank funders. This project was funded by Gradient Fund, which was supported by GSK and Novartis and registered by SAMRC. I also would like to acknowledge NITEC with student bursaries, and CHPC for the computational resources. I'm part of two Aurora Guild Core for AI and for GHA, and I would like to also acknowledge them. Thank you very much.
Thank you so much, Professor Ozlem, for this great talk. Really, it's great to see how much to correlate between the structure and the variants, and with the metabolism, actually correlate with the phenotype. And that's something amazing, because we need this type of in silico search, before to go for validation, you know, and looking in patient and sometimes for with functional studies. My question for you, you
Mhm.
did talk about some mutation that happening in the active site. This is for the NAT enzyme. And you're saying that the D112 arginine to N,
Mhm.
that's gave it like the most because it's happening in the active site, and it's giving the most important like changes in the structure compared to the one for example, the variant R64Q. It's giving some kind of changes, but not as important as in the active site. Now, my question for you, because you're saying even the R64 is like further away from the active site, but we are showing that because it's a dynamic, it's a continuity between the nuclear, the residues of the protein structure. It's making some changes in the conformation. Yeah,
Yes,
it can
local.
affect even the
Yes.
active site. How you can
Yes.
explain, please, that for us? When it's
Yeah,
far
this
away,
is this is called a dynamic allostery. It has been known since some time now. So allosteric effect can lead like drastic conformational changes in the protein, especially in dynamic proteins. But it can also cause local changes, which basically leads to the change the activity. This concept is known since I don't know, perhaps 20 years now. And what we are seeing is in pharmacogens, because they are not really very flexible dynamic proteins, but there are all these variations far from active site, which are leading to changes in metabolism. And it is basically due to this so-called dynamic allostery. It's a bit confusing because it's not dynamic actually. It's very locally changing the structure. So in 64, for instance, what we are seeing, it's changing the hydrogen bonding around that, which is leading the whole flexibility in changing the flexibility in domain one, which is affecting indirectly to the active site loop kind of things. So changing, perhaps it has no direct effect in the reaction like the 1 to 2, but it is affecting the mechanism of the protein by changing the active site shape or the communication or the flexibility. So
uh,
that's what's happening.
good. Do you have some experimental results, like just to validate the because this is models. Do you have some experimental, you
Well,
know,
we know that we have clinical results, right? I mean,
crystal structure?
we no, but we know that R64Q is actually a slow acetylator. We know that. So, and it is making sense because when we look at two rapid systems, we are not seeing that kind of change. We are hardly seeing any changes. But then when we look at compare these two rapid systems, which are behaving more or less the same, when we compare now to slow systems like R64Q, we started seeing quite a bit of changes. So I mean, we have clinical evidence.
Good,
So it
good.
is, yeah.
Okay. Now let's move for the cytochrome, the CYP3A4. There's a question. I will take the question first of all, and I will ask later if there's time. Can you explain in terms of the structure? This is Colidandara. Oh, good. Can you explain in terms of the structure of CYP3A4 what the enzyme seems to be able to metabolize most commonly used drugs?
So sorry, what is the question
Can
again?
you explain in terms of the structure of CYP3A4 what the enzyme seems to be able to metabolize most commonly used drugs? Which drug is specific to CYP3A4? Hello? I think we lost, yeah, we lost her. Okay. Let's see if she's back. Not yet, she's not here. Okay. Maybe just let me see. Okay, are there any more questions from the audience? No, she's not anymore back. Let's see. Nuzipo?
I think Prof is back. Ed Wilson, can you please unmute Prof Ozlem?
Where she is.
Hi, can you hear me?
Yes, we can hear you.
Yes, sorry, my laptop crashed again, but I'm using Rehema's laptop. So the question is how cytochrome 3A4 can accommodate different drugs? Was it the question?
Yes. Which one the most common?
Oh, this I don't know. Rehema, do you know?
Well, in my thinking, it's because the enzyme is highly flexible
Yeah,
and
no, but which drug says? But it basically metabolizes roughly 50% of the drugs, and this is basically due to the active site, which is more flexible and bigger than the other cytochromes.
Okay. Do you think this is happening with other cytochromes? Like because we know that, for example, the CYP2D6 also is highly polymorphic.
That's
And
right.
do you think it's the same thing? Like we can expect the same thing happening like CYP3A4, like with those different stars,
Yes.
variants. We'll see it's almost like the same, like with the modeling, we can find out that it's the same thing happening between
Yeah,
the phenotype and the structure.
Yes, we are actually looking at some other CYP enzymes, and we are in the process of writing CYP2D6 one actually. And in the channel behavior and things, we are seeing the similar things. So it's basically affecting, not him is not moving, the active site is staying more or less the same, but the channels are changing. The flexibility in certain loops are changing, and we are seeing more or less the same thing in CYP2D6. So in my opinion, after looking at number of them, we probably will be able to get a general idea how these variations would be affecting the protein.
Okay, yeah, that's great. That's very interesting, actually, to look in silico exactly and validate what's happening in patient, and try to correlate between both and between the phenotype in patient and in silico, like how we can, with the structure, we can observe that changes in the folding of the active sites or of the general folding of the enzyme or the protein structure. That's perfect. Okay. Are there any questions? Any more questions for Professor Bishop? Ozlem? No? If there's no any more questions, I want to thank you for the great talk. It's really amazing, to see this type of work, really like to validate what's going on in patient. And we are so happy to have you today and to have everybody actually in the webinar. And we hope to see you again in another webinar of African Pharmacogenomic Network. Okay, thank you so much.
Thanks a lot. And apologies for the technical challenges.
No worries. No, it's fine.
Okay.
Okay, then everybody, see you next webinar. Let's stay in touch. Okay, thank you.
Bye.
Okay, bye. Take care. Okay, bye.